Hey guys, I'm sorry that I can't be at school today. Killian still isn't feeling better, so I'm gonna stay home today. That way, I can be back at school on Tuesday. But if y'all have any questions in the meantime, let me know. I'm sorry I'm pushing back our quiz, but we will take it first thing on Wednesday, and then we'll review a little bit from 9.1 to 9.10, and then we'll move on to the next section of chapter 9. Um, so let's go ahead and get started. All right, so this is just your introduction. It's just going to go into, or this particular chapter is going to go into how traits are passed on from parents to offspring. And then, like now that there's technology that you can actually sequence genomes or like the entire genetic code of an individual or of an organism, people are, there's a lot, it's a very fast growing industry and science in order to build the knowledge base on like how can we actually look at the genes, isolate the genes, what genes on which chromosomes lead to whatever traits, and so on. Right. So this is just one of those main questions of the unit, which is are humans evolving? And so, depending upon, like what traits are present in the population and things like that, will determine if they are evolving. Right. And then the big ideas, we're going to go over Mendel's laws. We're going to discuss variations in Mendel's laws. We're going to discuss the chromosomal basis of inheritance. And then we're going to talk about sex chromosomes and sex linked genes. All right. So let's go ahead and get into Mendel's laws. Right, so the study of genetics has ancient roots. Right, Hippocrates' idea that pangenes travel from each part of an organism's body to the eggs or sperm is incorrect because the reproductive cells are not composed of particles from somatic or body cells, and that changes in somatic cells do not influence gametes or your sex cells. The blending hypothesis was rejected because it did not explain how traits that disappear in one generation can reappear in later generations. So these were two very early theories or ideas about how genes were passed from parent to offspring. And so, through the work of Gregor Mendel and through advancements building upon what Mendel taught us about how traits are inherited, it led to these being disproven. And so it's just another example of how science changes from, generation to generation, based on what information has been found, studied, and then, if it, once it turns in, or once it is considered a theory, it's the explanation for all of the situations that you see related to that phenomenon. All right. And so that's what leads us to Gregor Mendel. Right. So before we get into that, we have a checkpoint question. So it says, imagine you have two different birds of the same species, a female and a male. Female has a yellow beak and the male has a blue beak. And so it wants you to design a simple experiment to test the blending hypothesis. And so the blending hypothesis is when, if you were to cross the two birds, you would get a blend of the two colors, right, as a result. And so if we did an experiment, we would want to mate the yellow beaked female with the blue beaked male. And according to the blending hypothesis, we should get a bird with a green beak. But what we find is that there's going to be one trait that is dominant over the other. And so you'll see the dominant trait is expressed. All right. So the hypothesis for blending would be that there is a green beak. And then if the beak is not green, then you know that the hypothesis for blending was proven incorrect. Right. And so this is Hippocrates. So again, this is like, one of the, original people that, started asking questions like how are traits passed from one generation to the next. All right, so the science of genetics began in an abbey garden. An abbey is where monks live, and that was Gregor Mendel, he was a monk. The couple of definitions you have: heredity is the transmission of traits from one generation to the next. And then genetics, which is the scientific study of heredity, began with Gregor Mendel's experiments. A heritable feature that varies among individuals is called a character. All right, we know them right now as like variations, but they originally, what he called them was a character or a trait. Oh, okay. Hold on. I see what they're saying. All right. So a feature that varies among individuals is called a character. So, like eye color would be a character, but then the variant of the character would be the different eye colors that were there. So Mendel crossed pea plants and traced traits, which are variants of the character from generation to generation. And he hypothesized that there are alternative versions of genes or alleles, the units that determine heritable traits. Okay, so just a couple of important vocabulary words and the basis of what his beliefs were. All right, so it says describe the three generations of your own family using the terminology of a genetic cross. So you have P, F1, and F2. Right, so P would be generation would be your grandparents, and then they mated and they gave either your mom or your dad. And so they're going to be part of F1. And then, when they mated with either your dad or your mom, then that created you, which is going to be F2. Okay, so for example, I have my Momo and Papa, my Momo Jan and my Papa buddy. They're on my mom's side, so they're going to be the P generation. They had my mom, and she married my dad, so that's the F1 generation. And then they had me, my brother, and my sister, who are the F2 generation. All right, so this is our dude, Gregor Mendel. He was a monk. And so he did a lot of work within the garden in the abbey. This is the anatomy of the garden pea, pea flower. All right, so you have one petal is removed. So typically there's a petal like right here over the carpel, and so it's removed so you can see it better. And so you have your petals, you have your carpel, which contains the egg, and then you have your stamen, which releases the sperm containing pollen. And so that means that these flowers are reproducing sexually, because when the pollen is taken from one and put on the carpel of the other, then that's whenever they're going to start producing like seeds and flowers, flower buds and things like that. All right, so let's take a look at what he did with these peas. All right, so he removed the stamen of one parent. All right, and then he transferred the pollen, okay, from the white pea plant to the carpel. All right, so he got rid of the pollen created stamens on the purple flower, so there wouldn't be any accidental, cross pollination within the flower to make more purple offspring. So he got rid of the pollen on the purple one and took the pollen from the white one and put it on the carpel of the purple flower. And so when that happened, right, it led to the maturing, or the carpel maturing into a pod, right? So you can see the pea pod, all right? And then, all right, he planted the seeds from the pod, and then he observed the traits of offspring, right? So that would be our F1 generation. And so he found that if the parents were a purple one and a white one, that the F1 generation were all purple, okay? And so what does that tell you? All right, if you had to guess which one was dominant over the other, right, because the offspring are now all purple, you would lean towards the purple being the dominant trait, okay? And that's exactly what he found. Right, so by doing this exact same experiment, there's a couple of different traits that he was able to observe with these pea plants. And so we just saw the flower colors, right? But then he was also able to look at flower position. So they had axial is, which is coming from the stem, is dominant to terminal, which is like coming out from the very end, right? Seed color, yellow was dominant to green. Seed shape, round was dominant to wrinkled. Pod shaped, you had inflated versus constricted. Pod color was green is dominant, yellow is recessive. And then you had stem length, which was tall versus dwarf. Right? So he was able to do the exact same experiment over again with all of these other different traits. All right. And so let's see. All right. So he has 3 laws. Okay. And remember, laws are kind of like the lay of the land. Right. So this is what has been proven over and over and over again, kind of like the law of gravity. Right. So he, at first they developed, or they were hypotheses, and then he was able to test them over and over and over again. And then there have also been tested even in our generation, and even, way after Mandela passed. And the same, results were happening. So that's how they kind of became accepted as law. So he developed 4 hypotheses described below using modern terminology. All right. So the first one, there are alternative versions of genes called alleles that account for variations in inherited characters. Right. So you have your trait, right, which is the inherited character, and then you have your variations. Right. So, like, if the trait was, I don't know, pod color, okay, you can have a variation of green pod color, right, and you can have a variation of yellow pod color. All right. So, then you have like stem length, and you have your variations, tall versus dwarf. Right. And then for each character, an organism inherits 2 alleles of a gene, one from each parent. And so this kind of makes sense because we get half of our chromosomes from our mom, half of our chromosomes from our dad, and those alleles are found on those chromosomes. Right. An organism that has 2 identical alleles for a gene is said to be homozygous for that gene. The Latin root word homo does mean same. And then an organism that has 2 different alleles for a gene is said to be heterozygous for that gene. And hetero is a Latin prefix for different. Right? The third one is if 2 alleles of an inherited pair differ, then one determines the organism's appearance and is called the dominant allele, and the other has no noticeable effect on the organism's appearance and is called the recessive allele. And then you have a sperm or an egg carries only 1 allele for each inherited character because allele pairs separate or segregate from each other during the production of gametes. And we know that because they were, they line up in the middle on the metaphase plate, and then they're pulled apart during anaphase. Right. And so this statement is called the law of segregation. Right. And segregation just means that they're separating, and that they're separating randomly. Right. So then you can see where across that tracks 1 character. So this would be the character of flower color, and the 2 variations, right, are going to be purple flowers versus white flowers. Right. And these are true breeding plant parents. And so what that means is that this is going to be homozygous purple versus homozygous white. And now we just have to figure out which one's dominant versus which one's recessive. And so if we were to cross them, right, the F1 generation, they're going to have all purple plants, right? And so they're considered hybrids because they have 1 allele from the purple and we have 1 allele from the white, right? And now we're seeing which one's dominant, right? So because you had a cross between a purple and a white to make purple, right, then that means that that purple trait is dominant. So the going back up to the P generation, we would have like big P for purple and little p for white. This purple flower would donate a big P. This white flower would donate a little p. And so that would give you a heterozygous or hybrid big P, little p, which would be a purple flower. All right. And so then he wanted to know, well, what happens if I take some of these purple flowers that were made in the F1 generation, these heterozygous traits, and then figure out like if I crossbred them, what would happen. And so that gave us the F2 generation. So he fertilized two F1 plants, and that gave us our second generation. And so he got 75% of the plants were purple, and one fourth of the plants were white. All right. So, let me see. Can I pause? Okay. All right. So, Mendel's law of segregation explains the inheritance of a single character. So his hypothesis explains the 3-to-1 ratio observed in the F2 generation. The F1 hybrids all have big P, little p genotype, so that'll make them heterozygous. And then a Punnett square shows the 4 possible combinations of alleles that could occur when these gametes combine. So the checkpoint question for this one is, how can two plants with different genotypes for a particular inherited character be identical in phenotype? And so the reason for this is that you have homozygous dominant and then you have heterozygous. Just as long as they have one capital letter, it results in the dominant trait, right? So whenever he did the cross of the big P, little p, and then I'll fast forward, right? So we have a cross between a big P and a little p, right? So that gave us a big P, that would be purple, two big P, little p's, and you have a little p, little p. All right, so going back to our checkpoint question, how can two plants with different genotypes for a particular inherited character be identical in phenotype? So how is it that, you can have like big P, which is purple, and then big P, little p, which is purple, and it's because they have that dominant allele, okay, that makes them that dominant color. All right. So for this one, all right, in our P generation, the genetic makeup or alleles of the plant in this generation are purple flower, which is capital P, white flower, which is lowercase p, and then all gametes of the purple flowers are capital P, and then all the gametes in the white flower are little p or lowercase p. Right, and then in our F1 generation, right, they're all going to be hybrid, or heterozygous, which is big P, little p. So half of their gametes are big P, half of their gametes are little p. Right, and then we have in our F2 generation, we have the cross between two heterozygous individuals, one with a big P allele, one with a little p allele, right? And then so I always teach Punnett squares like this P will drop down, this one will come over, so that makes big P. This one will come over, and then the top one will drop down, big P, little p, right? This P will drop down, little p comes over, and that's big P, little p. And then you have little p. So this little p comes over, this one drops down. And so your phenotypic ratio, so phenotype is going to be the appearance, physical appearance, phenotype, right? So we have 3 purple and 1 white, right? And then for our genotypic ratio, genotype is just referring to the genetic makeup or the combination of alleles that it has, right? And so we have 1 big P that's made, 2 big P, little p's, and 1 little p, right? And so that is what is expressed in this picture as well. Right? Homologous chromosomes bear the alleles for each character. So every diploid cell has pairs of homologous chromosomes. The chromosomes in a homologous pair carry alleles of the same genes at the same locations. So the checkpoint question says, an individual is heterozygous, big B, little b, for a gene. According to the law of segregation, each gamete formed by this individual will either have a big B allele or the little b allele. Which step in the process of meiosis is the physical basis for the segregation of alleles? All right, and so the big B and the little b alleles are located at the same gene locus on the homologous chromosomes, which separate during meiosis 1 and are packaged in separate gametes during meiosis 2. All right, and so you have the separating that occurs in meiosis 1, and then again the separating that occurs to put them in individual gametes in meiosis 2. All right, and so this is just an example or an image of gene loci. Right, so this is the locations on the gene where different alleles occur. These are homologous chromosomes, one from one parent, one from the other parent. Right, and so the two homologous chromosomes show the gene loci for gene P, right, gene A, and gene B. All right, the chromosome with, let's see, a chromosome with the dominant allele, big B, has gene loci labeled. Right, so here we have our dominant allele. Right, and then capital P. All right, and then you have a lowercase a, right, and a lowercase a, right, and then you have a lowercase b. Okay, so in this position, this gene loci, comparing the homologous chromosomes, you have a big P allele and a big P allele. So because it's big P, that makes it homozygous dominant. Right, then for gene A, you have a little a and a little a, so that's homozygous recessive. Right, and then you have a big B and a little b, so that makes the genotype big B, little b. That's heterozygous, where you have one dominant allele and one recessive allele. All right, and then I will end this video and then pick up right here in the next one.